the Chief Medical Officer of the company. Pierluigi and Carlo, thank you for being with us today.
Thank you, Emanuela.
So maybe let's start with a brief overview of the company and its novel immune gene therapy platform for those investors who may not be as familiar.
Okay, thank you for that. So we are developing a cell gene therapy strategy to treat tumors, and our technology are based on the capability to control specific anti-tumor payload and to express within the tumor microenvironment this specific payload. And we are able to control the expression thanks to a microRNA downregulation. We are in the clinic. We are treating patients in GBM in a phase 1/2A. And now, after the IPO, we are fully financed until the end of 2024.
Great. So maybe we can start by discussing the timeline of your current programs and why is now the time to pay attention to Genenta.
Thanks, Emanuela. Yes, it is time to pay attention to Genenta because we are in a phase 1/2 clinical trial, and we are presenting at various different meetings at congresses some updates on our clinical study. It is important to also recognize what the Genenta may be able to offer in a very successful immuno-oncology field in cancer. And what I think is important is, and we are presenting to various meetings, is this novel approach to immunotherapy. And as we all know, we are really energized by the success of immuno-oncology in one particular type of cancer, which is the blood cancers, leukemia, lymphoma. What the Genenta technology is not to provide incremental value to what was achieved, but to open a completely new space, which is the space of solid tumor. And as you know, Emanuela, there is very little that was accomplished in the treatment of patients with solid tumor. So our founders, Professor Naldini, come up with the idea to develop a platform that, as Pierluigi said to you, will allow us to reach solid tumors, to deliver the treatment within the solid tumor, and to avoid systemic toxicity. And in addition, since we are targeting stem cells, to provide a long-lasting responses to the treatment. Where are we in the clinic? We are in a phase 1/2 in GBM. We can discuss why GBM later. And we already treated 15 patients in a classical gene therapy, cell therapy dose-ranging study, small number of patients in different cohorts. Up to today, we tested 15 patients, and we have 6 additional patients in 2 additional cohorts that will be done between now and the end of the year. That's where we are in the clinic today. And as I mentioned to you, for example, there's going to be a presentation at the AACR meeting very soon, where we're going to be providing some data from the ongoing study.
Perfect, thank you. So, you of course touched upon these on the rationale behind Genenta idea to going after solid tumors. I want to take a step back, and Genenta can claim a prominent experience and expertise among gene therapy platform, as the very first ex vivo gene therapy approved was developed by Genenta co-founder Professor Naldini. And it was for the treatment of a rare disease, but now, exactly as you said, Genenta is going after oncology. So what is the rationale behind the development of your leading asset, Interferon?
Yes. As you said, Genenta is, if you like, in a unique position in the field because our founders is the well-recognized father of ex vivo gene therapy. And as you just said, he was very, very successful in developing several different programs for monogenic rare diseases. However, throughout all this work, he acquired significant learning from the clinical as well as from the manufacturing side of ex vivo gene therapy. And all of this knowledge resides with Genenta. So what we are doing, we are learning and taking all the learning from the monogenic rare disease approach to a much broader field, which is the field of oncology. Obviously, we are talking about not a monogenic disease anymore, but a more complex series of events that will result in cancer. And as I mentioned before, we have the very strong foundation in how to generate the product, how to modify the stem cells, how to transfer the stem cells to the patient, how to monitor the cells that are derived from the stem cells throughout the duration of the study. All of this is a knowledge that is very relevant for what we want to do now in oncology, coming from the experience from the cell and gene therapy in rare diseases. So what I can tell you is that the ability to successfully and safely transduce cells with the lentivirus vector and the alpha interferon in humans is already being established. So we have data that show the safety and tolerability approach we are taking. We have data that already show that we can follow the progeny of cells that derive from the transplanted cells for up to 2 years from the transplant. So we have already demonstrated the proof of concept from a biological point of view in the ability to use this technology in cancer patients.
Yeah, maybe tell us a little bit more about Interferon and how it works.
Okay, so Interferon is called Interferon because TEMS is a population of monocytes that have been known to be preferentially localized within solid tumor. Interferon because we use in the interferon alpha, so that's the name Interferon. Now, there are three, in our opinion, major challenges that still remain in immuno-oncology. Number one is how to reach a solid tumor. Number two, how to avoid the systemic toxicity. And then finally, what we learn from the CAR-T in leukemia lymphoma patients is how to maintain a prolonged response in these patients. The durability of response seems to be still a challenge. Now, Professor Naldini and the co-investigators, they realized that perhaps using this population of TAMs that seems to migrating within the solid tumor will be a way to deliver a treatment to solid tumor. And they develop a very sophisticated technology to control the expression of the payload, in this case alpha interferon, only in the mature population of myeloid cells, which reside within the microenvironment of the tumor, avoiding the systemic toxicity that will be associated with the expression of the payload at all different stages of differentiation of the myeloid cell population. So we have a preclinical data, now clinical data, showing that indeed the systemic toxicity is not present in a treatment because, yes, we can measure the payload, the alpha interferon in the blood, but the level is very, very low, picogram levels, which is way below what could be a threshold associated with systemic toxicity. So that is already being accomplished. Now, you can ask why interferon alpha? As probably people remember, interferon alpha has been quite an effective treatment against solid tumor. The reason why it is not pretty much used in the clinic anymore is because of the systemic toxicity. And now with the ability to release the alpha interferon only within the microenvironment of the tumor, we can have the apoptotic lysis of tumor cells, which is the very well-known function of alpha interferon, and avoid the systemic toxicity because there is no level high enough systemically to potentially be associated with toxicity. So that's where we are. Now, regarding the last challenge, the durability of the response, we are not targeting the mature monocyte population. They will be going through few divisions and then die, but we are targeting the stem cells. So we are creating a factory of cells that will continuously produce these TAMs with alpha interferon.
Mm-hmm. So maybe a couple of questions related to this. So you mentioned you avoid in this way systemic toxicity. So I guess the first question is: Are you happy with what you see locally, and are you happy with the distribution you see in the around the tumor, in the tumor microenvironment? And the second question, I'll let you answer the first one.
Okay, so the first question, as you know, Emanuela, in order for us to obtain approval to test the product in humans, we have to provide the regulators with evidence of the biodistribution of your product. It's important because TAMPs are not only within the microenvironment of the tumor, but there are a few TAMPs that are also in endothelial cells, for example. So I'm happy to report that both in humans and in animals, the majority of the TAMPs that we transduce are within the solid tumor. Now, what we are seeing, now we have access to the tumor in GBM patients when the patient go into disease progression, because there is a second surgery, and therefore there is the ability for us to have access to the tumor. Now, remember, this is a dose-ranging study, so the patient in a very low dose cohorts are patients that some of them went into progression, and we had access to the tumor. So what we were able to see is the presence of TAMPs, so the TAMPs was within the tumor. We saw the alpha interferon release, and we saw also in some patients infiltration of CD8 T cells. All of this is replicating what was seen by Professor Naldini in the preclinical setting. Now, you may ask, okay, you see this biological evidence of activity of interferon, however, the patient went into progression. Now, the reason why we believe that it went into progression, because as I said before, we are talking about a very low dose of interferon, and we can make a correlation between what we see in the peripheral blood in terms of the progeny of cells derived from the transplanted one. And we can see that after 30 days after transplant, in a patient, we see 30% of the peripheral cells expressed in a vector copy number, which is the biomarker of the transplanted cells. However, over time, there is a decrease which remain above the threshold set up in a preclinical setting that we think is needed to show a potential efficacy. So in the dose ranging study where we are, and that's the reason why we amended the protocol to add 2 additional cohorts with a higher dose, we believe that going up in the dose, we're going to be able to see probably more clear evidence of the ability to modulate disease progression.
Okay, got it. So I guess you're getting this question all the time, but why did you choose a challenging indication as GBM and as your first clinical indication? And can you tell us maybe a little bit more about your expectation for these first human studies?
Yes, Emanuela, indeed, it is a very good, relevant question to ask, since the GBM is probably the most challenging cancer, with really still not an answer from a therapeutic point of view. There are 3 fundamental reasons why we believe that the GBM is an appropriate target for interferon. The first one is the biological evidence, with a numerous number of publications describing a myeloid population in the GBM patient in particular that seems to correlate with an induction of tolerance. In other words, in the inability of the immune system of patients with GBM to recognize and see the tumor. The second is the clinical evidence of this massive infiltration of myeloid cells within the tumor. The neurosurgeons are telling me, Carlo, this is very unusual to see such a massive infiltration of myeloid cells. This is exactly the population that Professor Naldini was able to convert from a pro-tumor to anti-tumor in the preclinical animal setting by modulating through the alpha interferon the microenvironment of the tumor. And then finally, we recognize that when you propose to test a treatment never used in humans before, in oncology, you're going to be required to go at the end of the line, meaning you're going to go in patient that fails one treatment after the other. Now, that in general is a problem because the patient is not in good conditions. But besides that, it's a significantly not a starter for us because the efficacy of Interferon does require a competent immune system. And the GBM patients are unique because, unfortunately, the disease is so aggressive, the patient actually die in 15, 16 months, and the tumor is only localized within the brain. So the immune system of patient with GBM is a competent immune system. It just is blind, it's not able to see the tumor.
Mm-hmm. Makes sense. So in terms of what are your expectations for this study, what would you be happy to see when you finish your first human study?
So the study, Emanuela, is a phase 1, phase 2 study, 2A study, and it was designed purposely to have a dose-ranging study, phase 1, and then an expansion of the safe and well-tolerated dose into a phase 2 part of the study. So by the end of this year, we should complete the phase 1 part and test 21 patients altogether. I expect to be able to use the highest dose, probably 4 million Interferon cells per kilo, in a phase 2 study that is under design and discussion. And the phase 2 study is going to be much larger, and we'll also have, it's still going to be open label because in gene therapy, it is impossible not to do an open label study. However, we will have a control arm of the study, now it's called synthetic control, where a patient population will be matched to the one undergoing treatment and will be used as a control, and the patient population will undergo standard of care. So that is something we plan to start in 2023, next year, probably end of the second quarter, when we have all the data from the phase 1 part of the study.
Got it. And could you describe your level of interactions with the regulator in Europe? And when you were saying you're planning to expand to phase 2 studies, are you planning for a global expansion? Should we expect the study to expand in the US, for example?
So the answer to the second part of your question is absolutely yes. If we are going to be able to have evidence of potential benefit, we will expand the study in US in addition to in Europe. Now, at the moment, the study is done in Italy. It's done in two institutes in Milano with the contribution of other centers in Italy. And we plan to do the phase 2 study in Italy at the moment. However, we already had a pre-IND meeting with the FDA to discuss our program, all aspects, the clinical data, the manufacturing strategy, the preclinical data, because we want to make sure that we are aligned with expectations from the regulator, not only in Italy or in Europe, but also with FDA. And I'm happy to report that it was a successful interaction with the FDA, and I don't perceive any potential issues from that point of view when Genenta will be ready to open a US IND study. Now, in addition, we have an advisory board that has membership from the US. And we have already established a working partnership with a group in Harvard, and they are very much interested in the technology that Genenta is developing. So at the moment, the activities are going to remain in Italy under the regulatory agency in Italy. But we already have interactions with the regulatory agency in the US and KOL in the US, and also manufacturing site in the US for a potential transfer of the manufacturing also in the US.
That's great to hear. So can you describe what are the opportunities for Interferon beyond glioblastoma? What is the time? And also, I meant to ask before, what is the time between apheresis and reinfusion, and how does this impact the application to other solid tumors beyond GBM?
Yeah, Emanuela, obviously you know the space because that is a very important question that you're asking regarding the manufacturing timelines. Now, in the GBM, the manufacturing timelines is not relevant because after the patient is enrolled in the study, we collected the stem cells, then the patient goes into 6 weeks of radiotherapy as a standard of care. And then we have plenty of time to make the product, and then when the patient is ready to treat the patients. That may not be the case in other patients with other cancers. And therefore, we are spending a significant amount of time in reducing as much as possible the release steps that are needed to release the product. And as you know, some of the testing require a significant amount of time. Adventitious aging testing require culture on the cells, and we're working with experts as well as regulators to find an appropriate compromise in terms of releasing a product that is sufficiently safe, even if it's not completely the all round of testing. And as we do right now, we do two-step approach. The first is quite quick, and then the second will provide the additional data after the product is already being used. Now, the difference regarding, for example, CAR-T is that our technical process of transducing the cells is very brief. It's only a few hours. And therefore, the technical cell therapy modification is very quick. What it takes longer is the release assays, as I just said.
Mm-hmm. I understand. You mentioned before, obviously there are, you can see advantages for the use of Interferon as compared to CAR-T, and the first one is the expansion to solid tumors, obviously. But you are also hypothesizing potential combinatory studies between Interferon and CAR-Ts. What is the rationale for that, and what would be other potential combination for Interferon?
Right. So the technology platform we are developing is allowing us to look at two parallel strategies. One is a standalone product, which is the Interferon, for example, where the agnostic nature of the product, which is not tumor-specific or antigen-specific, will allow us very quickly, after showing a proof of concept in one solid tumor, to really expand to a very broad range of solid tumor where the TAMs are responsible for creating this immune tolerance. So as a standalone product, you can imagine how widely you can use this product. In addition, Professor Naldini already has really interesting preclinical data showing the ability to synergize in a safe and efficacious way other immuno-oncology product using Interferon, which to me is not an alternative, but is a parallel way to provide value to patients with cancer. We know that CAR-T can be effective, but we also know CAR-T has been not an effective treatment for solid tumor. Now, Naldini is going to publish in a very important paper data where he showed that actually a CAR-T that alone seems to be not as effective in a solid tumor when it's in presence of Interferon, the ability, the efficacy is significantly increased. Now, why is that? Most likely because Interferon is modifying the microenvironment of the tumor, which allow the CAR-T to benefit from the whole immunological response to the tumor. And I'm very excited about seeing those data that you can imagine could be of interest to many companies that have a product in development and also product on the market with a limited penetration of the market because the limited potential value that is viewed from payers, patients, and physicians. And so quite exciting about seeing the potential combination data coming out.
Yeah, absolutely. That's very exciting. So you touched upon this before. We've seen during the past several years how manufacturing readiness has been certainly a key element, especially in the context of late stage developments and approval from regulators. Can you tell us more about Genenta's work and plans around manufacturing?
Absolutely. Number one, we are lucky. The manufacturing of Genenta's product is the same company that manufactured the product for GlaxoSmithKline, for Orchard Pharmaceutical, and therefore is a CMO that's been approved by regulators both in the US as well as in Europe, not only to manufacture a product for development, but also for a marketed product. The standard is much higher in there. So we are quite confident that the way we're making the product today is good and is reassuring. Now, your question is, okay, however, how are you going to expand your clinical trial, expand the reach into different markets, et cetera, et cetera. So there are two considerations. The first one is that if we were discussing this maybe 5-6 years ago, you will see the manufacturing readiness was essential. So startups were ready to start manufacturing their own product even before having any view whether the product could be successful in the clinic. Why? Because there was no market there. There was no availability to manufacture cell and gene therapy, and therefore you need to build your own. Mm-hmm. Now we are in very different space. The cell and gene therapy, it is a market, is a big market, and there are plenty of players that are competing to provide services. So they really basically tell us, okay, we can wait until making a decision whether we need to invest in manufacturing our own product or not, and are going to allow us not to make the mistake to invest too early. However, we are also aware that it's going to take time. In order for us to transfer the technology, it's going to take time in order to book the slots in the potential CMO. So we already established conversation with our own current AGC, is the name of the company that's making our product today, for potentially manufacturing the product ex-US, ex-Europe in the US. But we're also talking to different other CMO in the US with a particular interest in the cell therapy approach. And the whole space is really growing very, very quickly with trying to manufacture the product in a much more sophisticated way, in a closure environment, in an environment that could be more physically located within clinical centers and so forth and so on. So I think the space is moving in the right direction. We are comfortable where we are today, and we are now waiting to establish our pattern for expansions when it's appropriate. So we feel that we're okay there now.
Makes sense. Thank you for that. And so just in closing, you have an upcoming presentation at the AACR. We are looking forward to that. Maybe you can set our expectation for that presentation, and also our expectation for the next 12 months. And maybe this last question I can ask to Pierluigi.
Yes. So for the AACR, Bernard Gentner is one of the founders of the company, is going to be presenting the update on the clinical trials. So he's going to basically present what I just mentioned to you, the safety data on the 15 patients that we have, and also the biological evidence of the presence of Interferon. Particularly, he's going to be presenting some interesting data on the tumor from a few of the patients that we were able to have access to. And it's going to present some very sophisticated analysis using single-cell microRNA sequencing, as well as T-cell receptor sequencing, to really show the presence of Interferon, the presence of the alpha interferon, and the infiltration of CD8 cells. These data that are very, very interesting from the point of view of understanding the mechanism of action of Interferon in humans. And I'm looking forward to see how the presentation is going to be received, and ready and happy to follow up and having conversation with the community that is trying to solve a very challenging problems. And I also want to emphasize, Emanuela, that we should be very thankful for all the patients and families that are willing to enter in our studies. And I want to keep focusing that what we're trying to do is to help patients that don't have at the moment any answer from a treatment point of view.
Absolutely. We cannot say that enough. You're right. So, Pierluigi, maybe, as I said before, in closing, what should investors focus on in the next 6 to 12 months?
First of all, as we mentioned at the beginning of our conversation, it's important, especially in this market, to have raised enough money to move on with our clinical study. And we can perform and complete the phase 1/2A, as Carlo mentioned, and one inflection point could be at the end of this year when we release this data. During that, we have AACR and other scientific congresses. And in the first part of 2023, we will start the expansion in the phase 2. These are the most important things that on which we are focused as a team and as a company in the next 12 months.
Great. Thank you very much, Pierluigi and Carlo. This is very exciting, and we look forward to follow the development of Genenta in the future. Hopefully, our next conference will be one that we can hold in person rather than virtually. But in the meantime, we are very grateful for your flexibility and your presence online this year. So thank you again from the HC Wainwright team.
Thank you, Emanuela.